Hello again. Today we have a big topic, multiple sequence alignments, and we'll discuss some tools and practicalities related to file handling and file formats.
Hello, this is a test before starting. Hello, this is a test before starting. Good morning. This is the fourth stream lecture of our bioinformatics workshop. Today, Dr. Tovanen will talk about multiple sequence alignments, which is used in countless bioinformatics applications. So it is worth to pay special attention to this lecture. Okay, before we start, I would like to reflect a bit on the video consultation yesterday evening. There were several students coming and we have clarified questions about the assignments, which is the very goal of this opportunity. We have stayed until after 4 o'clock, but we were far from exceeding our 25 people limit. So today I encourage you to come early if you have questions. The session will be again open this afternoon from 3:30 and we will stay at least until 4, or if we need more time, as long as there are students with questions. So come to ask. The sessions are purely for your benefit. But now let's dive into the details of multiple sequence alignments.
Hello again. Today we have a big topic, multiple sequence alignments, and we'll discuss some tools and practicalities related to file handling and file formats. So what do we mean by alignment? It's actually two common meanings. One of them is the process of adding gaps into sequences, DNA or proteins, so that we get homologous sites stacked in columns. In naive terms, this would be maximizing identity. And the other meaning is the result of the process of alignment. Alignment is the form of sequences with dashes so that we have them aligned. But anyway, to be less naive, the optimized quantity is more like the sum of matches, mismatches, and gaps, which we take from substitution scores, substitution matrix scores, and gap penalties as discussed yesterday. And again, there are many algorithms for arriving at an optimal algorithm. Pairwise alignments are a bore, just two sequences. We don't know if the gaps are quite correct, and the conserved site, an identity, we don't really know if it's truly conserved and constrained in evolution or just something that didn't have time to mutate. And a non-identity could represent a highly variable position, and we have two samples of the variance there, or maybe it is a conserved site and we are just unlucky so that the other one of the sequences happens to have a different residue. So if n equals 2, we don't get really good statistics. But a multiple sequence alignment is obviously better because we have more data for each column in the alignment. We have a better view of sequence conservation. We can really see if every sequence out of 20 has the same residue in column 60, so it is probably an important residue and truly conserved. And conserved in biology means that there is a selection pressure against changing that place. There will be consequences in fitness and viability if we break that place in a protein or DNA. And then MSAs are useful because we can extract sequence profiles, frequency matrices out of the aligned data. And these profiles would be good models for a protein family so that we can identify new family members by matching them against the profiles. And furthermore, three-dimensional modeling is facilitated by multiple sequence alignments, and also MSAs are kind of supportive for evaluating which parts in the model are reliable. So the conserved residues are probably also less mobile in their three-dimensional position. And furthermore, we have phylogenetic inference, which is nowadays more often done based on sequence alignments than on bone morphology, etc. Obviously, in dinosaurs, we don't have intact DNA, so we have to be happy with the with the traditional phylogenetics, but in molecular phylogenetics, each column in an alignment is one character in phylogenetics. And this is just an example of a phylogenetic tree, which is from a publication done together with Chaba and others, which showed that viral poxvirus CA-like sequences are descendants of CA13. We couldn't figure out what was the host organism when this jump happened, but anyway, the source protein is obvious there. Now, regarding MSA algorithms, this is tricky because we can't really do the full computation. Fully optimal MSAs can be done for toy problems like 5 sequences, but larger than that, you have to use heuristics and approximations. And there are 3 classes of algorithms: progressive, iterative, and probabilistic. The progressive algorithms are going out of fashion because they just can't handle larger amounts of sequences, and with lots of genomes which we have nowadays, they are getting just too slow. So these iterative and probabilistic are better classes, and we will be using algorithms called Clustal Omega or softwares called Clustal Omega and Muscle. But before going to the algorithm, maybe even more important than figuring out the parameters is your data. The selection of sequences is, in my opinion, the single most important step for getting a good quality alignment. If you have sequences which are kind of outliers in the bunch, less than 25% sequence identity, the algorithms can't handle them correctly in every case. So you may get misalignments, and that's a problem. And sequences of very different lengths with long insertions in between might throw the algorithm off the rails. So having more uniform data is always a big help. So observing your first alignment and then removing kind of outlier sequences and realigning is part of the game, which you just have to accept. So you should be doing visual inspection of your alignment, or if you are going to big projects like aligning 50,000 different viral strains, so you should develop programmatic tools which are able to tell you any outliers in the alignment. But for the cases we are handling, so visual works fine. And in real data taken from genomes of species which are not very well investigated, so that we don't have proteins or we don't have RNA-seq data, you have to predict the protein coding genes based on proteins of other species, human, mouse, zebrafish, such model organisms that have, and often mistakes happen in the predictions. There might be some exons which are not correctly predicted. There might be missing data, misassembled genomes, so that one exon is just not where it's supposed to be. And again, we'll see an example of quality control, how we can identify kind of suspicious sequences. And if we have alternative splicing, we should only include one isoform in the sequence if our goal is comparison of different species instead of comparing the isoforms to each other. And so all of the sequences should represent the same isoform, the canonical isoform, or the Apris P1 principal isoform. We'll talk about this when we go to ensemble. Or if we are looking at proteins, so we should nitpick that we are looking at the translations of these isoforms. But the idea is that we should have the same set of homologous exons that code for the proteins you study. If you have alternative exons, the exons are not homologous, they are from different places of the gene. They tend to be aligned to the same place where they are in the protein, but they are not homologous and your inferences are not correct. And likewise, if you have your sequences coming from a blast search result set, you shouldn't take sequences which have high e-values like 0.5 or 1, because there is no certainty that they are homologous. And if you don't take my word for that, so you should believe Gandalf. So this is like the 11th command for bioinformaticians. Don't force alignment of unrelated sequences. And now to repeat the best algorithms and some of the pros and cons, we have Clustal Omega, which is another incarnation in a long series of algorithm actually originating from Ireland. Clustal Omega is fast and accurate, and it scales well into large amounts of sequences. There's another one called Muscle, which is still very fast and accurate, but then the other programs in the list suffer from slowness, so that when you are going to hundreds of sequences, you are not getting your results very quickly. MAFT, in fact, is a collection of programs. Some of them are fast and not that accurate, and the slow ones are really good in best in the rankings, but I wouldn't be using them in daily work because the single alignment might take half an hour to run. And the bold facing here is indicating that we have them available in the package CView, which we are going to use in this course. Now let's take a short tour in MSA file formats. And actually we are only going to be looking at FASTA, multiple sequence files, and Clustal format files. There are lots of multiple sequence alignment formats, and luckily there are conversion websites. So if you do phylogenetics, your program might be requiring a specific file format like Nexus, and then you can go to a site like this MyHits, which gives you the possibility to interchange between the formats. But anyway, FASTA and Clustal are something you should be able to recognize just by looking at the file, so that you know what you tell your program to treat it as, and you give it a right name in case your program expects a specific name. And this is a multiple sequence FASTA protein file with 3 sequences. And the names for the sequences would be the first word up to the space, and the rest is just my personal notes, the GI numbers from NCBI, which I used to produce these sequences. And the sequences in unaligned FASTA most of the time are of unequal length, but this is fine because we are going to align them. And then we have aligned FASTA, in which the alignment program has added the dashes to represent the gaps. And now the sequences are of equal length, and if you are going to use these sequences further as aligned sequences, so they should keep the equal length. So be careful not to delete accidentally some of those dashes. And you should not be using MS Word for handling these sequence files, because the dashes will lead into line breaks going strangely when these are treated as hyphens that divide your hyphenated words. And also the MS Word line breaks in general might do funny things, so that your sequence name might end up into your data. And I've seen so many cases of your alignment programs just refusing to handle the data if it's been through MS Word. So find a proper text editor like Notepad++ to do your sequence editing if you need to use manual editing to add or remove sequences. This is a good format for removing and adding sequences, because you can just take take one block and it's there. But then in cluster format we have the sequences in a visually understandable format so that we actually see that we have a conserved site here, and we have some marks to indicate full conservation and partial conservation. And because the sequences have to be split into lengths that fit in your page or screen, so this means that the same block of sequence names is repeated over and over, and if you want to delete a single sequence, you might need to delete 10 different lines so that each line of that one sequence is removed. Don't do that. Rather do a conversion into fast A, remove your sequence and go ahead using it. Now, cluster comes in two different formats. The previous one is without sequence numbers in the end. So these numbers indicate the number within the sequences, which is the last character in that line. And you can see that some sequences which start only in the second block have 0 there. And now the numbered cluster format is sometimes refused. There are programs which just don't recognize cluster with numbers because it's a new thing that's been around only for 15 years or so, and not every program has had time to update their definitions. But anyway, important things to note when you are collecting your sequences to prepare for alignment. So take care that all the sequence names are all different up to the first space. So if you have CA6 human and CA6 mouse, if you have a space there, so you will have two sequences called CA6, but CA6 underscore human will be just fine on side of CA6 underscore mouse. And once your sequences are aligned, so all sequences need to be of the same length. So if you do some accidental mouse moves so that you are taking out a single character from one of your sequences and you try to process the alignment further for phylogenetic tree or whatever, so it will be invalidated by changes in the length. When you have a cluster formatted file, there are some things you need to include if you're copy-pasting the full alignment from somewhere. So one of them is the first line, which has to have cluster as the first word. It doesn't matter what are the numbers, cluster W, cluster O are just fine there. And then the other thing is if you have this consensus line indicating the similarities and identities, the consensus line has to be with every block, including the last one, even if it is just with spaces. But then cluster without consensus line is another format which is valid. Okay, so be prepared to run into some troubles with your multi-sequence files. You might encounter a problem with doesn't like your data, but luckily the program we are going to use, CView, is pretty tolerant, so that's really nice. Now, regarding the visualization of your MSAs, if you want to include example alignments in your thesis or for a publication, so it has to be of a good quality. Screen prints are not that nice, so you need to have vector fonts, something that produces a rich text format file or a PDF. And what we have is CView, which has a PDF output, and the lucky thing is that it also has the alignment algorithms built in, and even phylogenetic tree algorithms if you want to follow up in that direction later. And I recommend the drag and drop interface so that you just take your MSA file from a folder and drop it in the main window of CView. You can do it through the File Open menu dialog, but then you have to choose which format of a file you want to open, and for CView to find the file in that folder, it requires that your cluster files are named .aln, and the FASTA format files can be named .fasta, .fa, .fst. And there are multiple other formats, but let's just stick with the two most familiar ones. Okay, now we are going to take a quick demo on slides. I'm not going to show you CView in practice on a shared screen. I have ready-made parts which I'll introduce, but you really should download CView. It is a good software, and you will need it in your MSA Mini project to make the alignments. Of course, if you are familiar with the EBI alignment servers, there's nothing to stop you from using your familiar tools, but for those of us who might be new to MSAs, so learning CView has a smaller threshold than going through the web servers. Okay, I have a gene tree for CA6 taken from Ensemble, and it is not of great quality. There are some clearly erratic features, and we'll take a look at them to see how to judge an MSA, what kind of features you should look at an MSA of yours once you have one of your own. And I have the gene tree file available in Moodle, so you can repeat this practice with CView if you wish. This is the starting point, a clearly rough MSA that has some strange gaps, some incomplete sequences and whatnot. So I have more details in the next few slides. So to show you the bad spots, here's a sequence in which we have clearly something missing. There's nothing to align very well to these spots, and there is some extra, probably because a gene prediction program just wanted to create something that is of the same length as CA6. Looking more carefully, this pin is repeated here, so we might have some problems in the genome assembly so that the actual content of this exon has been moved out, or maybe we have something that is improperly predicted. But anyway, we can see it's not correct, so we want to get rid of this sequence. Okay, here we have a large part missing from the beginning, so we would take it out. Here it seems we have misalignment, so that this R should clearly be the second amino acid. And then this M is way most popular for a start site, as you can see, and all the sequences which have something else here are probably mispredicted, so that the algorithm just takes the longest possible open reading frame with an M in the start, and that's it. But this wouldn't work. This is a secreted protein, and this is not a signal peptide, whereas here we have the sequence starting with the signal peptide. But we don't need to care much about this. We can just ignore them, trim the whole alignment, or just start our phylogenetic analysis starting from this position in the MSA. But if you just want to have a cleaner alignment, so feel free to throw out the very long one. Again, here we have missing some sequence, and in some sequences we have long regions of X's, which are a result of some more intelligent gene prediction algorithms which realize that they can't find the exon coding for this part of the protein, but because it should be there, they are adding the same length of X's as they have in the model organism, probably human. But again, this is a problem for the phylogenetics, so you would want to get rid of it. And now, for quality control in CView to execute the removal of sequences judged bad by the criteria we saw, so you can just select with control clicks all the sequence names that you want removed, and then going to edit, delete sequences. They will be gone. But this misaligned sequence, even if it doesn't look good, it probably can be aligned to look better. So we'll see pretty soon how to do realignment. Okay, we had one sequence which had an insertion in this region, and with the single sequence with the insertion gone, we have now columns that contain nothing but gaps. So going to edit and deleting gap only sites would put the ends of this long gap back together so that we have a nice clean alignment there. Now regarding alignment, so here is an align menu, and if you go to alignment options, you can choose whether you are using clustal omega or muscle. Setting parameters for them is tricky, so we are not going to talk about it. And there's also a chance of defining whichever alignment algorithm you install in your own computer to be used here, but we'll skip that too. But anyway, we'll be using clustal omega, which was what I had set in the alignment options, and choosing to align all the sequences will be realigned. And you can see that the sequence, which is 2 sequences above this long sequence, now has this misaligned sequence nicely tucked into the gap. So that was clearly a problem in the original alignment for whatever reason. Maybe there were other sequences that just were misleading how this one sequence was aligned. Then looking at the C-terminus of these proteins, this is pretty messy. We don't really know if some of these sequences are incorrect. Maybe they are non-homologous, so that they are coming from exons of different origins, or maybe some of them are just mispredicted, and it seems that some of these ends might be missing. Most probably every CA6 should have something there. But anyway, again, we can just ignore this problem and decide that we go later into some other tools that will trim the alignment from this point onward, or just decide, if we do phylogenetics, that this is the last column we are going to use. Now, regarding saving your alignment in a clean format, there is pretty easy to use tools here. First, you have to go to PDF options and check that your paper size is okay, and depending on the size of your alignment, you might want to put it into a fairly small font so that more of it fits on one page. If it's a small one, it's fine to have a more readable font. You can choose between color and black and white for PDF, and block size we'll see in the next slide. But after you set your options, so pressing prepare PDF will create the file. And here's an example, and the block size here of 10 means that they insert a space after every 10 residues. And also, if you choose a block size of 30, it would mean in this case that you would only get 30 residues per line, probably, because now we can fit 50 sequence characters, 5 blocks, but you couldn't fit 2 blocks of 30. But anyway, this is really sharp. So if we look at the screen capture, which we have here, so this is not nice quality. Magnifying the picture a little bit reveals that this is not really good quality. It's okay for your quick notes for your lab notebook or private notes before you prepare your final figures, but for the actual figures you really want the PDF. Well, in a way I'm cheating because this is a screen print from a PDF, but you can see that the PDF is not breaking into pixels when you magnify it, and when you send the PDF to your publisher or include a PDF in a document, so that will be a good way to do that. Okay, let's move to one more topic of MSA visualization. We have problems if we have lots of sequences in the alignment. You can fit 30 or 50 sequences on one page pretty easily, but when your blocks are becoming multi-page blocks, you really can't compare the sequences on one page and the next because the columns are not visible at once. So you want to condense the information in some way to get large alignments into a tangible format, and sequence logos are really nice. The way sequence logos are created is that the algorithm observes each column separately and it calculates the frequencies of each amino acid in that column, and there is a quantity called information content which it measures the noise in the communication. It comes from communication theory way back from 1948, or maybe signal analysis. But anyway, the more agreement you have in the column, the higher its information content. And the information content is given as the height of each stack of letters for that position. So here we can see that we have a fully conserved tryptophan, a W, in position 26. And then in the next position we have a partially conserved site which has threonines and serines, and now the heights of T and S represent the frequencies of T and S in that column. And you can see that the information content is going fairly low when we have a mix of 5 or more sequence characters. And what is more, this sequence logo uses conventional colors for acidic and basic, nonpolar and polar residues, so that they are really informative. After you've studied them even just for 5 minutes, you start to get the hang of it. And most importantly, so you can fit easily like 10 of these 50 residue blocks of sequence logo on one page, and that would give you a 500 residue protein, the full alignment, the information of the full alignment in one visual grasp. And now, as the last thing for this presentation, I will make a short demo of one sequence logo creation tool, Berkeley Web Logo, and then we'll talk a little bit about your MSA project. Now for the web logo demo we are going to use another file which is in Moodle. It's called 6, 9, 12, 14 proteins aligned, and the numbers, you guessed it, they refer to different carbonic anhydrases. And before going to web logo, I'm going to check that the sequences are indeed properly aligned, and I'm going to just drop the file into this main window of CView, and you may see that we indeed have many sequences, and the alignment does look clean. We get columns of a single color to show that we really have conserved blocks aligned, and there are some differences in the end, but we don't need to worry about that for now. And there's also a very long tail in the N-terminus for CA9 of some species. But this is good enough for us. At least the catalytic domains are properly aligned. So we can be happy with that. So let's move to our web browser window. And we can open Berkeley web logo from Moodle. And here we go. We go to creating logos, and I'll just paste the sequence data here. I have the file open in editor, and I just select all and copy, and I'll go to pasting there in the window and create web logo after we selected the parameters. Let's go for 50 stacks per line and skip the fine print and create web logo. Now we'll see that the beginning has lots of very low letters and very narrow stacks because we don't have the data. We probably want to be starting this logo from position about 140. And we might want to stop it at 420, so let's go back. Logo range 140 to 420. And now this is becoming more readable. And now we have error bars which are measuring the uncertainty of the information content which we might not want there. And also we have a statistical thing which takes into account the background frequencies of amino acids, so that a conserved tryptophan will have a higher column than a fully conserved leucine. And if you don't want that behavior, so we can just go for no adjustment of composition and get something that resembles the old style. And now the colors are different from the version 2 web logos I was showing, which we could also change in the settings, but this shows you how easy it is to get these web logos. So instead of getting crazy in looking at 30 lines per block, so we just get each column represented by a single stack of characters, and we can easily see where the highly conserved sites are, and this QSP and this HXH and this H and this TTPP are actually important parts of the catalytic site, as you'd expect with high conservation. As a final topic regarding MSAs, we'll be talking about the mini project you will be carrying out using MSA tools. As indicated in the syllabus and in our first meeting, so we have a project with peer review. This is using a special Moodle function which allows distributing the reports of all of you to be reviewed by a number of peer students, and we are going to limit it to just doing 2 evaluations in addition to your own work. But the reason we do such a complicated and seemingly an exercise of a lot of work is that MSAs are probably one of the most useful skills that is good for doing phylogenetics, doing 3-dimensional modeling, and visualizing sequence conservation in protein models. And there are so many things for which the MSAs are a requirement, so it's very basic. It's almost like pipetting for laboratory skills, so it needs to be practiced. And we want you to learn to use MSA tools so that you can actually take a set of sequences and produce a reliable MSA. And also, if you are going to do a sequence related project later this spring and you already know your supervisor, so we have been given a set of sequences that would be of their interest, and maybe you can do sequence collecting already for a further project. So we will have a look at that in Moodle as well. And as rewards, so you will be getting maximum of 6 points for this whole thing, 4 points for the actual workshop and 2 points for the evaluations. And there's a fairly detailed instruction set, a couple of pages in PDF in Moodle, in which you find lots of practical instruction for this. But in brief, the project involves collecting protein sequences starting by a BLAST search from a specific query sequence. And if you don't have a supervisor and a designated protein, so there will be another set of sequences suggested for you to do your MSAs on in the end of the document. Still not there as I'm recording this. And then you do a multiple sequence alignment of those proteins and then do visualization. Use CView to produce a colored PDF and use web logo to create a sequence logo, or maybe a fragment of a sequence logo if your sequences are really long. But just to show that you know how to use this technique. And the idea is that you would be reporting your methods in a way that would allow your peer reviewers to see that you've actually done this properly, and maybe you will learn some new tricks looking at the way that others did the same work. And there's also going to be a small essay regarding an MSA algorithm. We'll have a look at that in a minute. And hopefully you might be able to get some insights into the sequences you aligned and observe where you have conservation. Maybe compare your alignment with information from UniProt if any functional residues are reported, so you might be able to pick them up. But anyway, because of the technology in Moodle, so this has to be done synchronously so that all the reports are submitted by Sunday evening Irish time, Sunday midnight rather. And I hope you'll be able to do this mostly by Friday so that you'll have a free weekend anyway. But there's no way to extend the deadline, so if you run short of time, so you just have to cut your goals and leave out some parts of the task. So submit whatever you have done by Sunday evening to get at least some points, because it's not possible to make it better and submit it one day later. And then for the peer evaluation, there will be a 3-day period as well. I'll open it for evaluation on Monday morning. And in fact, no, I don't want to encourage you to do an all-nighter, so let's keep the deadline here. And then on Monday morning, about 6 a.m. of Irish time, I will start the evaluation period. Now, the tasks and evaluation have been set to a total of 60 points, but I'll do the rescaling after the evaluations are there and the averages have been calculated. So there's the essay part, your description of one algorithm, and you get points for very easy things like just finding your sequences, for making an alignment of whatever kind, and then points for the colored version, which would be submitted as a separate file if you can't include a PDF, the sequence logos, and then almost half of your points would be of documenting and reflecting your work. Reflection is pretty important for remembering and learning more deeply what you have been doing. So I really want you to do that, and you are rewarded for that. The evaluation instructions are telling you that the kind of thinking of your results, thinking of your process will be really important. So just copy pasting the results and not writing too much about it will give you about 35 points out of the 60. So take care to document how you did it, so that if I read your work, I could reproduce what you did. Your input sequences are good to put there as an appendix, as a FASTA file. So that is a very simple and unambiguous documentation, especially if you have retained your accession numbers from the database. And this evaluation is really important for your learning, because reading another person's work is kind of revising the things that have been done, but also seeing them from a different angle. And we have 4 different algorithms to write your essays on, so it would be really bad luck if you are reading three essays of the same algorithm. There's probably going to be a couple of different ones. But you'll be getting 60 points and 30 points maximum of raw points. Here the evaluation points might be decreasing slightly if your evaluations are not agreeing well with the other evaluators, but that's like a very minor subtraction. We are going to use a relaxed scale on this scaling, on this evaluating of your evaluations. And in the end, I'll be dividing the points by 15 so that you will get this to the scale of 6 points. Now let's have a look at the PDF document. So this includes all kinds of encouragement, reminder of the deadlines, and then talk about these focus proteins. Now, to give good alignments, I'm suggesting that you would be filtering the sequences so that you would choose something in the range of 30 to 60% identity. If you just take the top hits and they happen to be more than 95% identical, your sequence logo will be just all characters of identical heights, and you don't get to estimate what are the important parts based on the conservation profile. But I did some random checks of some of the suggested proteins, and many of them are extremely strongly conserved, so that there would be 200 animal proteins in the range of 97 to 100% identity. That doesn't really make a good alignment. So if your supervisor is giving you all such proteins, you just have to revert to the CA option that is given in the end if no good targets come up. Also, there were some sequences that had homologs in invertebrates and in plants, but in those cases the query coverage was maybe just 25%. You could then tweak these instructions so that you are not requiring 95% query coverage, but instead require 25% and trim your original query sequence so that you are only studying one domain in the original sequence, which has homologs in other kinds of organisms, so that you get your variance into the sequences. But anyway, this is a little bit tricky, and I hope you will find a good target quickly enough. Maybe you can discuss this thing with your supervisors and mentors so that you get better ideas collectively. And if you realize that, okay, this sequence is a bummer, so feel free to write in the forum that no, we can't really use this FBOX42 protein because it is too highly similar. I forget the exact name, apologies, that was one I tested. But anyway, the suggestion is to take some sequences of kind of average level of conservation and then add the original query sequence so that you can use that as a reference point in the alignment. And as said, I'm recommending CView, but there's no problem in using the EBI MSA web servers just as well if that is what you're used to doing. And it would be good if you don't take the default of CView, which is Clustal Omega. For variability between the reports, it would be good if you take an actual coin or a six-sided die or whatever, or do a mental throw of a die to get a decision on which algorithm to use. But anyway, for MSA theory, I have assigned you an algorithm based on the last digit of the day of the birth. So I'm born the 8th of September, so I would be choosing MAFT based on 8. And the sources you might want to use for writing this MSA theory summary, I'm suggesting half a page to one full page, but maybe half a page is more realistic, considering the time constraints and the level we are expecting. You can find some documentation in the EBI alignment servers, or you can do searches in PubMed or Google, find the website of the developers of the algorithm that will lead you to more information. But anyway, I have some ideas, some concepts here that are related to the nuts and bolts of the algorithm, so trying to make sense of this will at least get you superficially familiar to what is happening when you run an MSA. And I have some ideas of what kinds of things you could watch for in the interpretation. You can try to figure out if there are some structurally important residues. Maybe we can use your MSA in the three-dimensional structure exercises later on for doing visualizations. But anyway, it's all here, and this is copied directly from the instructions that are going to be in the evaluation phase. When you go there, you'll be giving points on different aspects, and the aspects are as indicated here from A to G. So 8 different aspects, 7 different aspects there. So it's going to be a lot of work, but in my previous courses, so students are saying in the end of the course that this was the best exercises because you really had to put your skills into some real use, and doing the write-up really made you think about what you were doing and learning it properly. We are going to have another live meeting in Big Blue Button in the afternoon today. So if anything is unclear by that time in the instructions, so feel free to drop by and get clarifications, or if you can't make it, so write in the forum, so we'll send written answers to anything that was not that clear.